Yes, that's fine. Yeah, that's fine.
Do you want to wait a couple of minutes on Dr. Wyvolyn? This is just back. So, and then there's a reason. So, okay, we're ready for our next talk. This is Dr. Webby's student, Wyvolyn, and we are very excited to hear her presentation.
My experiment was done on the solvent effect on the extraction efficiency of antioxidants. Solvents are the medium used to extract antioxidants from out of fruits and vegetables. Antioxidants help prevent cell damage caused by oxidants in our biological systems. And when I say the term extraction, I mean the process of getting the antioxidants from outside of the fruits and vegetables. My motivation for doing this experiment was because there are many different types of solvents used to extract antioxidants from out of fruits and vegetables. Previous studies have been done using these solvents in different forms and for different periods of time. So my basis for the study was to attempt to develop a more efficient way of extracting these antioxidants. My objective was to determine the solvent effect on the extraction of antioxidants and polyphenols, and in doing that, determine which solvent had the highest extraction efficiency. In my experiment, I used red peppers and garlic, and these are an example of what they would look like. To begin my experiment, I purchased my vegetables from the local market. They were washed with deionized water and they were weighed and measured. The red peppers, I removed the inedible parts and they were chopped up and blended. The garlic was peeled to reveal the naked inside, and those were also blended as well. These samples were then freeze-dried in a lyophilizer. To extract the solvent, I used 1 gram of the sample and mixed it with 25 milliliters of the solvent. The solution was shaken overnight, and then they were vacuum filtered. The shaking procedure was done twice, and the filtrate was used to analyze the sample. For my analysis, I used 2 methods. I used the FRAP method and the Follinger-Koppel method.
Yes, so we show yeah, just go ahead. Yeah, just exit out of that. It's on the other side. It said there's a problem with this drive, but it looks like it wasn't.
Yes, that's fine. Yeah, that's fine.
Do you want to wait a couple of minutes on Dr. Wyvolyn? This is just back. So, and then there's a reason. So, okay, we're ready for our next talk. This is Dr. Webby's student, Wyvolyn, and we are very excited to hear her presentation.
My experiment was done on the solvent effect on the extraction efficiency of antioxidants. Solvents are the medium used to extract antioxidants from out of fruits and vegetables. Antioxidants help prevent cell damage caused by oxidants in our biological systems. And when I say the term extraction, I mean the process of getting the antioxidants from outside of the fruits and vegetables. My motivation for doing this experiment was because there are many different types of solvents used to extract antioxidants from out of fruits and vegetables. Previous studies have been done using these solvents in different forms and for different periods of time. So my basis for the study was to attempt to develop a more efficient way of extracting these antioxidants. My objective was to determine the solvent effect on the extraction of antioxidants and polyphenols, and in doing that, determine which solvent had the highest extraction efficiency. In my experiment, I used red peppers and garlic, and these are an example of what they would look like. To begin my experiment, I purchased my vegetables from the local market. They were washed with deionized water, and they were weighed and measured. The red peppers, I removed the inedible parts, and they were chopped up and blended. The garlic was peeled to reveal the naked inside, and those were also blended as well. These samples were then freeze-dried in a lyophilizer. To extract the solvent, I used 1 gram of the sample and mixed it with 25 milliliters of the solvent. The solution was shaken overnight, and then they were vacuum filtered. The shaking procedure was done twice, and the filtrate was used to analyze the sample. For my analysis, I used 2 methods. I used the FRAP method and the Follinger-Cockle method. With the FRAP method, the reagents were prepared with 2.5 milliliters of TPTZ and 2.5 milliliters of iron 3 chloride, as well as 25 milliliters of the acetic acid buffer. That solution, I used 2,600 microliters of that solution with 400 microliters of distilled water, and then 100 microliters of the sample to do the analysis on the UV-Vis spectrophotometer at 595 nanometers. For the Follinger-Cockle method, and for the FRAP method, I use iron 2 as the standard, but for the Follinger-Cockle method, I use the gallic acid as my standard. I used 150 microliters of the sample and mixed it with 150 microliters of the Follinger-Cockle reagent, and that was set at room temperature for 5 minutes. After 5 minutes, I mixed 300 microliters of distilled deionized water and then 2,400 microliters of sodium carbonate, and that was vortexed and then placed in a dark area for 30 minutes. After 30 minutes, then it was analyzed at 765 nanometers on the UV-Vis spectrometer. This is a relative result of my analysis. It shows the equivalent concentration of the iron 2 standard, and as you can see, the 40% aqueous methanol showed the highest result. And when I say 40%, I mean 40%. 40 microliters of methanol and then 60 microliters of water. And this also shows the result for a triplicate extraction. This shows the result for the extraction of antioxidants on the red peppers from the ethanol sample. As you can see, the 80% aqueous ethanol gave the highest results. And then this one shows the extraction of antioxidants used by the acetone on the red peppers. And as you can see, the 60% and 40% were relatively close, but the 40% gave the highest extraction of antioxidants from the red peppers. And then this graph shows the result of all the red peppers, and this is for one extraction. The red pepper with the methanol gave the highest extraction for the antioxidants. This is the result for all the extraction. This is the result for all the solvents used with the garlic sample. And as you can see, the water gave the highest extraction of antioxidants from the sample, and this is from one single extraction. And the previous one was also used with one extraction. This is a comparison of both the red pepper and the garlic samples. With both of the samples, water gave the highest extraction concentration for the antioxidants. This shows the relative graph for the phologicoccal extraction of the antioxidants. I used the samples that had the highest concentration of antioxidants found from the previous sample in the FRAP method. So I used the methanol 40%, the ethanol 80%, and the acetone 40%. And with the phologicoccal, it showed that the ethanol 80% gave the highest concentration for polyphenols. In my conclusions, I found that the aqueous organic solvents for the 40% methanol, the 80% ethanol, and the 40% acetone gave the highest yield for concentration of antioxidants in the sample. However, water did yield the highest concentration for antioxidants with a single extraction. Overall, 40% aqueous methanol did yield the highest concentration of antioxidants with a triplicate extraction. My findings further indicated that 80% aqueous ethanol gave the highest concentration of polyphenols with the red peppers. But with the garlic samples, the solvents used in our samples were not really suitable. And I want to thank Jacksonville State University, JSU Department of Physical and Earth Science, JSU Faculty Research Grant, and Dr. Webby. Thank you.
Question.
Yes.
What does it mean when the extraction measure is negative, like on a couple of the garlics?
Okay, when I use the standards for that, we use an equation to measure how much was extracted for it. It kind of showed that not much was extracted at all, so that's why it kind of went to the negative side.
So it's more a measurement error?
Yes, ma'am.
Other questions? Dr. Webby, would you like to add anything to the study and your student? We started without you, sorry.
That's okay. That's my mistake, not yours. Basically, when you go into literature, you want to extract total polyphenols or antioxidants. There is so many solvents, so many ways, and so the idea was: is there a standardized way, a much more efficient process of if I know I'm getting vegetables and I need total polyphenols, how do I go? So this is why we were using polar solvents, but we were putting water in them to give them, that aqueous, characteristic, and actually that adds to their ability to dissolve. And so, yeah, this is work in progress. And one of the things we do also is we might have to add some acids, like acetic acid, to the solvents to prevent oxidation of your polyphenols or antioxidants, because that happens as they sit over time, because we were doing this for 24 hours. So that's the idea behind this. And she's using those two techniques just to quantify the antioxidants or the polyphenols. They're the most commonly used techniques.
Other questions?
On your methanol, your ethanol extractions, you're saying the aqueous seem to do best on that one-time fruit.
Yes, sir.
The polyphenols that I understand, I would think they would be soluble in your solvents. So is there a reason we think we're not getting them out, or we think maybe they're changing before or something going on?
I think they're getting oxidized.
Okay, so the method, so under those harsh solvent conditions,
Yes.
we're changing this chemical, okay.
And that's the reason why we might use some, acetic acid.
It's interesting the different percentages work differently. That's kind of a neat feature. I was wondering if that meant that there was something like, somehow the polyphenols, because you did freeze dry the samples first, right?
Yes.
And so that was 1 gram of freeze-dried material you didn't put your solvent in. Well, I'm assuming it was ground up plant material, so it's got all the cellulose, it's got all the other features in there. I was wondering, maybe something's binding up the phenols, the solvents can't get through, the aqueous helps soften it up, and then kind of pops it out.
Yeah.
I don't know.
Anything else for Wyvolyn? Okay, let's thank our speaker again. And our next talk will begin in about 4 minutes.
Difference in the aqueous,
Yes.
that would be wrong, you're looking for that magic percent.
Yes. And see the thing is,
So she did say the tryptophan was getting more and more out.
yeah. Again,
No one's died.
I've probably seen very
The one with the
I remember the face. With the time I'll get it. Yes.
And they are just, it's the first semester when you are actually doing job more like bring them in because they are our residents.
So we went for our schedule. It gets us to have some time.
Yeah, actually you are kind of, helping for teaching and because you met them teachers. I didn't know.
Yeah, it's
Kind of follow the procedure.
yeah.
Yes.
Once you get the orientation, you watch other things.
That you have to have all the time. Observe. Don't blow up, right? Yes. I was not sure what they are doing. Really? Yes. It's in order.
Until I walked in, I didn't want to come. I didn't want to get numbers. Well, we have some
You did great.
Thank you for warning me. I would have been like,
Okay, I think we're ready for our next talk. So if everyone would gravitate back towards, grab a snack and get it to go. Come on, back up for the next talk. I think we are ready to begin. Dr. Rayburn is going to introduce the student.
Okay, those of you who were here earlier, I guess there aren't too many there, but this is a third student who came and approached me, I guess at the end of the summer. We got together. Actually, it's this fall.
Yeah.
So that's fall. About doing a project, and this one is going to be a little bit different. We're looking at, she was interested in working with salamanders, and I had a student who had previously done some work with chromium and salamanders and got his master's, and wanted to work with amphibians and was interested in eutrophication, and then maybe I've added a chemical into this, and we'll see how things go with that. But working with salamanders, and Kayla's been just getting started, so this is sort of a proposal with just the tiniest bit of preliminary data that she's got. So she's been, matter of fact, some of this stuff is active, ongoing as we speak.
Yeah, and this is just kind of what I want to do my thesis on, so I'm not used to using that. This is awkward. Okay, so this is the title of my proposal. It's a proposal for investigating the potential environmental effects of nitrate, ammonia, phosphate, and perchlorate on spotted salamanders. I gotta watch that thing. Okay, so I get asked a lot when I'm talking about what I want to do. They're like, what is eutrophication? And so I decided for this, I would just quote my favorite limnologist here, Wetzel. And he said it's a multifaceted term associated with human impact on aquatic ecosystems, increased productivity, that's a major one, and simplification of biotic communities, which results in reduced adaptation and stability. So the adaptation, that's another big one. It mainly occurs from nitrogen and phosphate, but there's other factors that come into play for that. So human impact has been well documented, but it can also occur naturally over geological time. So I just want to show you guys this picture here. So this is natural eutrophication that happens, like I said, over time. You have an unproductive lake here, and it takes thousands of years just to get to a mesotrophic lake, which is just kind of in the middle. And then it takes hundreds of years to go from that to this, so a really long time. But for the anthropogenic causes of eutrophication, you have a oligotrophic lake up here, and it goes to eutrophic in a matter of decades. And these are some of the causes here. I'm mainly interested in runoff and fertilizers and pesticides. Water body size is something that I have to take into consideration because I'm interested in studying ephemeral ponds here. It's been generally accepted that small water bodies exhibit different characteristics than larger ones, but I actually have data like myself that I've collected. I don't have it up here, but